Hey, what's up guys? My name's Efren. I'm going to be talking about this lovely, beautiful, helix sighting thing and just how mesmerizing it is, and you can really tell just by looking at it. I wish I could make it spin, but I couldn't. But you might get the, you could, let me just break it down for you. It's pretty much one of the most complicated molecules that exist, and potentially the most important one. It's so complex that we didn't even know for sure what it looked like until about 60 years ago. It's so, awesome that if you just took all of it from one of our cells and entangled it, it would be taller than I am. That's crazy. Now consider that there are about probably 50 trillion cells in my body right now, laid end to end. The DNA in those cells would probably stretch to the sun not once, but 600 times. Is your mind blown yet? Well, probably. But here's the thing. It's always happening. And you're probably making one right now. Of course you know what I'm talking about. It's deoxyribonucleic acid. Ribonucleic acid. Known to its friends as DNA. DNA is what stores our genetic instructions and information that programs all of the cell's activities. It's about a 6 billion letter code that provides the assembly instructions for everything that you are, and it does pretty much the same thing for every other living thing. Gonna go out on a limb here and assume you're a human being, in which case you are every cell in your body, somatic cell rather, right now has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed tightly together with proteins in the nucleus of the cell. The DNA is a nucleic acid. In DNA, these small units are called nucleotides. Linked in together, and you have yourself a polynucleotide. Now, before we actually put these little tiny parts together to build a DNA molecule like some microscopic piece of IKEA furniture, let's just take a look at what makes up each nucleotide. Because I know that we take all these classes, a semester at a time. So you might have forgotten. Here we go again. In chromosomal mutation, which is what we're going to be talking about today. Chromosomal mutation. So means that something like AGGTCCGTA does not mean the same as TCCGTTTGA, right? Human chromosome 1 is the largest human of all of our chromosomes. Contains a single molecule of DNA with 247 million base pairs. 247 million. If you printed all the letters in chromosome 1 into a book, it'd be about 200,000 pages long. And each of your somatic cells has 46 DNA molecules tightly packed together into its nucleus. That's one for each of your chromosomes. Put 46 molecules together, and we're talking about roughly 6 billion base pairs in every cell. The longest book I've ever read is right here. It's about 1,000 pages long, right? If we were to fill it with our DNA sequence, we'd need about 10,000 of them to fit our entire genome. Scumbag. DNA's replication gets it wrong about once every 10 billion nucleotides. But don't think your body didn't have an app for that, because it turns out DNA polymerase can also proofread in a sense, removing nucleotides from the end of the strand when they discover the mismatched base, because the last thing you want is an A when it should have been a G. Considering how tightly packed DNA is into each one of our cells, it's honestly amazing that more mistakes don't happen. But they do, and remember, we're talking about millions of miles worth of stuff inside us. And so that, my friends, is kind of the prelude to my chromosomal mutations assignment today. I need to educate you on this. You need to know this. This happens every day. It's really common. When little kids say they want to grow up and they want to be a scientist, here's what they really mean. They want to blow stuff up in the laboratory. They want to get bitten by radioactive monkeys, which will turn them to terrifying humanoid battle monkeys and make they want to make like a chicken with eyeballs on its wings or something like that, or like flies with fangs. But most of the times, scientists, they don't get to do that stuff. Like, you know, you may blow something up, but it's either going to be in a really controlled setting or it's going to be an accident, in which case it's going to be bad, like blood-stained bad. But, you know, there's an amazing new discipline. It's called evolutionary development biology. I don't mean to go on a tangent about this, but you might get to make a fly with eyeballs on its butt, you know, or even a chicken with teeth, but no battle monkeys. No battle monkeys at all. So how do they figure out, you know, how to do this stuff in their genes and genetic? Like, is it a mutation? Yeah, it could be. It could be an insertion, perhaps. But here is the geneticist's job. The geneticist's job is to pretty much identify chromosomal mutations, why they occur, and take a deeper look. So chromosomal mutation research today, geneticists look for unusual features that affect 1 in the 100,000 people of a population, typically referred to by primary care doctors because they don't know as much of these genetic mutations. So rule of thumb that geneticists have for these doctors is 2 unusual features which will warrant such a referral. Features such as physical problems, anomalies, health problems, learning difficulties, challenging behavior of some sort. And that will warrant the geneticist to come out and, you know, observe, speculate. And it's kind of the boring, monotonous part of being a geneticist in terms of chromosomal mutation, perhaps, because you really are more of a spectator and you can just document stuff, and, you know, you're going to start to feel like, you know, Mendel and all those pea plants. But really, like, if you want to get into the interesting stuff, you could start working with a one of those Evo Debo type of biologist that makes, you know, flies with, you know, eyes on the. But it's complicated, and they want the geneticists that know the chromosomal mutations. Why? Because there's this thing called developmental regulatory genes, which their job is to just tell you when to work and how much to work. So you have these like Hox genes, for example, that pretty much they kick into place whenever you are in the embryo, and you're going to be developing. So it's like they say, hey, make a mouth over here and make the anus on this side, and that's what these regulatory genes are. Because most of the times when we think about you know, proteins that are in the genes that are going to code for things, we think of like an enzyme protein or enzyme or protein that are going to be like the ones that say, hey, your ankles are going to be fat or something like that. And it's like, no, you don't. No. You have these genes that are pretty much, their job is to turn things on and off. They activate genes. They put body parts together. That put the body parts together. They don't tell them how to do it, but they just tell them when and if it's time to get to work. And since they're pretty much the ones calling the plays, they're pretty cool. I mean, they're kind of like the architects. And once they know the master scheme, they tell their subcontractors to do it. But of course, they're not going to do any of the construction because that's way below them, right? So there's tiers. You know, you have your first tier regulatory genes, then you have your second tier subcontractor genes, your third and fourth, and so on. And these all influence whenever there is some sort of mutation that occurs. So chromosomal mutation, you're looking really in depth because you need to know all this stuff. And you think, oh, they're just documenting, like these geneticists are just documenting what the patient looks like and what he's doing. No, they really are trying to put 2 and 2 together. So you've got your basics that you have to master, like passionately. Like you just, it has to be imprinted in the back of your head. Like you need to know every step of the replication and the DNA, and you need to know, you know, regulatory genes and background genetic information in order to really study chromosomal mutation today. Because in cracking the code, geneticists are limited. They have some obstacles. Geneticists, what they can do is give a prognosis of what's going on and possibly, you know, have it create a early intervention for further development, you know, such as medication stuff, perhaps. But what they're working with is the different type of chromosomal mutations that are happening, and all in depth. So you have chromosomal rearrangements, you have aneuploids, anuepliody, polyploids, and that one was the one that aneuploidy rather was the one I was starting to talk about, because an aneuploidy, the number of chromosomes is altered. In other words, like one or more individual chromosomes are added, and this is the wild card that I was talking about, you know, in terms of background stuff, because an aberrant chromosome number has been recognized as a common characteristic of cancer cells for more than 100 years, and it has been suggested to cause tumorigenesis nearly just as long. However, this proposal had remained untested due to the difficulty of selectively generating aneuploidy without causing other damage. But we've been doing it recently. In heterozygous animals which develop whole chromosome aneuploidy, which promote tumorigenesis in some contexts and inhibit it in others. These findings confirm that aneuploidy can act oncogenically and reveal a previously unsuspected role for aneuploidy as a tumor suppressor, and that is indeed taking all that information of regulatory genes and how, you know, genes are. I mean, not necessarily regulatory, but the intricate system that is happening in that chromosomal mutation, and aneuploidy is that wild card, because in that sense, whenever it is positive, that is a mutation that you might want to induce at any given moment if you ever encounter a cancer that could be suppressed. Furthermore, there's a couple of more of mutations that we're going to talk about. Chromosome rearrangement, what that entails, consequences, certain cases, and polyploids.